The topic taken for today is animal classification. Animals, though they appear different, they exhibit a fundamental similarity in their basic structure. Then, the similarities of different animals can be seen in their cells, symmetry, body layers, body cavities, different types of systems, and also embryology. Now let us see how the similarity exists between different organisms. First one, levels of organization. We know that all the animals are multicellular. Nowadays, phylum Protozoa is removed from the animal kingdom and it is included under Protista, kingdom Protista. So all the animals included under the animal kingdom are multicellular. Then, in some animals, the cells are not organized into tissues. It means the body organization is at cellular level. The cellular level body organization is seen in phylum Porifera, to which sponges belong to. So these multicellular animals are without any tissues, and these are also the lowest animals in the animal kingdom. The next level of organization is tissue level. A tissue is a group of cells often similar in structure and origin and perform a specialized function. There are 4 types of tissues in the entire animal kingdom. They are epithelial tissues, connective tissues, muscle tissues, and nerve tissues. So in the next phylum, that is phylum Coelenterata or phylum Cnidaria, and also in phylum Ctenophora, cells are organized into tissues. So we find tissue grade of organization in phylum Coelenterata and phylum Ctenophora. Then, organ level. Tissues group together and form organs. And organ level of organization is seen in phylum Platyhelminthes and phylum Aschelminthes. Then the next level of organization is organ system level. Organs make up a variety of systems. For example, digestive system, nervous system, excretory system, reproductive system, like that. This is called organ system level. All the phyla, beginning with phylum Annelida, belong to organ system level of organization. These are phylum Annelida, phylum Arthropoda, phylum Mollusca, phylum Echinodermata, phylum Hemichordata, and phylum Chordata. Then, protostomes and deuterostomes. During embryonic development, what happens is the gastrula has a cavity known as gastrocele. It opens to the outside by means of an opening known as a blastopore. In some animals, the blastopore becomes the mouth, and in others, it becomes the anus. The animals in which the blastopore becomes mouth are known as protostomes. In the case of deuterostomes, blastopore becomes the anus and mouth appears as a secondary opening. Deuteros means secondary, stoma means opening. So in deuterostomes, the blastopore becomes anus and mouth appears as a secondary opening. In this way, all the animals are divided into protostomes and deuterostomes. Phylum or phyla Porifera, phylum Cnidaria or Coelenterata, phylum Ctenophora, phylum Platyhelminthes, phylum Nemathelminthes or Aschelminthes, phylum Annelida, phylum Arthropoda, and phylum Mollusca. All these phyla come under protostomes. phylum Echinodermata, phylum Hemichordata, and phylum Chordata come under deuterostomes. In this way, all the animals are divided into two groups, protostomes and deuterostomes. Then coming to the blood vascular system, it is open type in some animals and closed type in other animals. In the type of closed blood circulation, what happens? Blood never leaves the blood vessels. It flows only through blood vessels continuously. And the closed type of blood vessel system is seen in phylum Annelida except leeches. Then gastropod mollusks and chordates. In all, and the open type of blood vessel system is present in leeches belonging to phylum Annelida, arthropods, mollusks, echinoderms, and hemichordates. In the case of open type of blood vascular system, the blood flows through blood vessels for some distance, then it comes out and flows through open spaces. That's why it is known as open type of blood vessel system. These open spaces are known as sinuses. Then another important level of organization is symmetry. Symmetry means arrangement of body parts along the principal axis or the long axis of the animal. If the body parts are arranged symmetrically along the long axis or principal axis, it is known as, we can say there is symmetry. If the body parts are not arranged systematically along the principal axis, it is known as asymmetry. Then symmetry is again many types. The first one is spherical symmetry. Spherical symmetry is seen in spherical animals or animals that look like a ball, like volvox belonging to phylum Protozoa. So in the case of spherical symmetry, what happens is they have many axes and many radii, and the animal can be divided into two equal halves along any radius or along any plane. Radial symmetry. Here, the animals have anterior and posterior ends, but dorsal and ventral surfaces are not formed. The body shape is either cylindrical or umbrella-like. In these animals, the body parts are arranged along and also around the principal axis. So these radial symmetrical animals can produce identical halves along the principal axis. Identical halves are also known as mirror images. Mirror images means you take our hands. If you put like this, they become mirror images. This hand is mirror image of this hand. Like that, we get mirror images or identical halves, which are also known as antimirrors if the animal is symmetrical. Then bilateral symmetry. In the case of bilateral symmetry, what happens is there is only one principal axis, that is the anterior-posterior axis. These animals have an anterior end, posterior end, dorsal surface, ventral surface, and also lateral surfaces. These animals are divided into equal halves only on one plane, that is sagittal plane. Sagittal plane means a cut made through mid-dorsoventral direction. So if a cut is made on the mid-ventral dorsal direction, it is known as sagittal plane, and bilateral symmetrical animals produce identical halves only on the sagittal plane. Then, from phylum Platyhelminthes onwards, all the animals exhibit bilateral symmetry with a few exceptions. Those exceptions are gastropod mollusks and echinoderms. Radial symmetry is seen in phylum Coelenterata, phylum Ctenophora, and also some sponges. Then coming to the next system, biradial symmetry. In the case of biradial symmetry, some body parts are arranged on radial plan and others are arranged on bilateral plan. And this type of symmetry is seen in ctenophores. Next, pentamerous radial symmetry. Pentamerous radial symmetry is seen only in adult echinoderms. In adult echinoderms, the larvae are bilateral symmetrical, but the adults, because of their sedentary habit, they have developed a pentamerous radial symmetry. This is known as pentamerous radial symmetry because all the body parts are arranged on 5 radii. This is a modified type of radial symmetry, you can call. Then, asymmetry. Some animals have lost the symmetry or they cannot be produced into or cut into equal halves. These are some of the sponges and also gastropod mollusks. The next level of organization is diploblastic or triploblastic. In the animals, the cells are arranged into 3 body layers or germinal layers. These are known as ectoderm, endoderm and mesoderm. Ectoderm is the first germinal layer, endoderm is the second germinal layer, and the mesoderm is the third germinal layer. If 2 germinal layers are present, they are known as diploblastic animals. For example, in phylum Coelenterata or Cnidaria and Ctenophora, only 2 germinal layers are present. So they are known as diploblastic animals. These 2 germinal layers are ectoderm and endoderm. In all other animal phyla, that is from phylum Platyhelminths onwards, all 3 germinal layers are present and they are ectoderm, endoderm and mesoderm. In this way, animals are divided into diploblastic or triploblastic. Then, the coelom. Coelom is the body cavity appeared or it is formed in the mesoderm. So, the coelom has 3 characteristics. That is, it is a cavity formed in the mesoderm. It lies between the body wall and the gut. It is also lined by coelomic epithelium derived from mesoderm. Then, coelom is again 2 types, schizocoelom and enterocoelom. Schizocoelom is formed due to splitting of the mesoderm, and schizocoelom is present in phylum Annelida, phylum Arthropoda, phylum Mollusca, etc. The second one is enterocoelous coelom. Enteron means primitive gut. In these animals, coelom appears as outpocketings from the primitive gut or enteron. This type of coelom is present in phylum Echinodermata, phylum Hemichordata and phylum Chordata. Then, in some animals, we also come across pseudocoelom or false coelom. It is not derived from mesoderm. Actually, the pseudocoelom is the remnant of blastocele or segmentation cavity present in the embryonic stage called blastula. Usually, the blastocele disappears, but in some animals like Ascalminthis or roundworms, there is a well-developed pseudocoelom derived from the segmentation cavity. If there is no body cavity, they are known as acoelomates. Then, segmentation or metamerism. The bodies of some animals are divided serially into segments or metameres. If the body is divided into segments, it is known as segmentation or metamerism. In the entire animal kingdom, only 3 phyla exhibit segmentation. They are phylum Annelida, phylum Arthropoda and phylum Chordata. In phylum Chordata, the metameric segmentation is clearly exhibited during embryonic development. In adult animals, it disappears to a great extent. Then, there are also some animals that exhibit pseudometamerism or false metamerism. The example is tapeworms belonging to class Cystoda of phylum Platyhelminths. In the case of pseudo-coelomates, body is divided into metameter segments, but they are not true segments. Then, notochord. Notochord is a solid skeletal structure derived from the mesoderm and present in all the embryonic stages of chordates. In the vertebrate animals, what happens is the notochord is replaced by a vertebral column. So if animals have a notochord during their developmental stages or at any stage of their life, they are called chordates. All other animals are known as non-chordates. In this way, the animals are broadly divided into chordates and non-chordates. Then, here I have given a broad outline of classification, animal classification. So, animal kingdom is broadly divided into two subkingdoms. These are known as parazoa and eumetazoa. In parazoa, tissue grade of body organization is absent. That means the body organization is at cellular level. Phylum Porifera belongs to subkingdom parazoa. Then, the second subkingdom is eumetazoa. Eumetazoa means true metazoans, true multicellular animals. In these animals, cells are organized into tissues. Then, eumetazoa is divided into two grades, radiata or diploblastica and triploblastica or bilateria. Radiata means these animals have radial symmetry, which I have explained just now. Radiata is also known as diploblastica because in these animals only two germinal layers, that is ectoderm and endoderm, are present. Then, phyla Coelenterata or phylum Coelenterata and phylum Ctenophora belong to this grade. Other grade is Triploblastica. Triploblastica means they have all the three germinal layers, ectoderm, endoderm and mesoderm. They also exhibit bilateral symmetry. That's why it got the name bilateria. Then, the grade bilateria is divided into three divisions: acoelomata, pseudocoelomata and coelomata. Acoelomata means animals without a body cavity or without a true coelom. Phylum Platyhelminthes belongs to division acoelomata. Then pseudocoelomata. Pseudocoelomata means they have a body cavity like cavity, but it is not true body cavity. Already I have told you, in pseudocoelomates, the body cavity is nothing but the remnant of blastocele or segmentation cavity. Because of the presence of pseudocoelom, this division got the name pseudocoelomata and the phylum is Aschelminthes. The third division is coelomata. In these animals, a true coelom is present. That's why it got the name coelomata. I have told you, true coelom has three characteristics. It is a cavity formed in the mesoderm. It is a cavity lying between the body wall and gut, and this cavity is lined by coelomic epithelium derived from the mesoderm. Then coelomata is divided into two subdivisions, schizocoelomata and enterocoelomata. In the case of schizocoelomata, the body cavity is formed due to splitting of mesoderm. Scission means split. Phylum Annelida, Arthropoda and Mollusca belong to schizocoelomata. In the case of enterocoelomata, the coelom appears in the form of outpocketings from the primitive gut or enteron. That's why it got the name enterocoelomata. Enterocoelom is present in phylum Echinodermata, phylum Hemichordata and Chordata. In this way, we find various levels of organization in the animal kingdom. Thank you.